So thank you so much, Muhammad, for this opportunity. And for those who might be joining us for the first time, I would like to start by introducing myself. So my name is Olfat Ahmad, and I got trained in pediatric oncology in Jordan. Then I got also trained in genomics at Oxford University and then the German Cancer Research Center. It's my pleasure to speak to you today on behalf of the Molecular Neuropathology Outreach Consortium, which, as you know, is an initiative of the German Cancer Research Center together with the University Hospital of Heidelberg. Before we start with this talk about inside the methylation workflow from tissue to report for non-lab professionals, allow me please to start with two quick disclosures. Molecular pathologists, technicians, and bioinformaticians, please forgive what we are about to present. This talk is intentionally made light. Secondly, please allow me to acknowledge that the Molecular Neuropathology Outreach Consortium has received in-kind support from Illumina as well as Heidelberg Epignostics. And without further ado, let me walk you through the roadmap of this session. First, we will define who is the targeted audience so that you know if this talk targets you or not. Secondly, what is the concept of DNA methylation? Then we'll touch on infinium methylation arrays and how this concept and technology was utilized to build the Heidelberg classifier. Then the comprehensive overview of methylation EPIC version 2 workflow from the sample to report, which is the core of our talk today. And finally, I will remind you of what my colleague Jan has presented last time regarding t-SNE plots and UMAPs. So if you're wondering whether this talk targets you or not, then probably you are in the right place if you've been recently engaged in methylation discussions and you've been hearing terms like IDAT files, CPGs, t-SNE plots, copy number variations, and you're really curious to know the correct order of the process and how these steps are connected and what's really going on in the lab or in your institution. Also, if you work in a lab and you're considering implementing methylation in your institution, especially if you are in a low resource setting and you want a practical overview of the real workflow behind the whole process. So let's start with the concept of DNA methylation. Methylation is basically the addition of methyl group to the cytosine base of the DNA. As you know, DNA strand is composed of 4 different bases, the adenosine, cytosine, guanine, and thymine, together with the backbone of ribose, sugar, and phosphate group. In higher eukaryotes, like humans, methylation occurs almost exclusively at CPG sites, and this is the first term that we would stop at. So when we say CPG site, it simply refers to a cytosine and a guanine that occur next to each other, and the P stands for the phosphate group here, simply. So methylation affects gene expression because basically methylation leads to condensation of the chromatin, and it hides transcription factors in a way and decreases gene expression. So methylated areas would be quieter in a way, and unmethylated would be more active. So if DNA was an instruction book for the cell, then you could say that methylation, together with other epigenetic marks, act as the sticky notes, which simply help regulate which part to read and which part to skip. And this explains actually how diverse tissue specificities can be derived from the same genome sequence. So if you look at the same DNA sequence with different methylation patterns, those differentiate into different tissues. Genome-wide DNA methylation profiling acts as a fingerprint then or memory for cellular lineage, making it well suited for classification of a tumor sample based on the cell of origin. And please focus here on the word genome-wide. So we are talking about profiling genome-wide of several locations across the genome, almost 1 million locations nowadays. So this takes me to the Infinium methylation epic array, which is the most popular tool nowadays for methylation analysis. If you look at this bead chip, which some of you are familiar with, it's composed of 8 wells. So it accommodates 8 samples. And this is the major factor that determines the turnaround time in your lab. And this is why you might be hearing lab people telling you that we are still batching samples. Because basically, if they run the bead chip for a single sample, then the cost of the bead chip will be wasted for one sample instead of 8, for example. If you look actually at a small sample section of the bead chip, it includes millions of beads. And each bead is basically coated with hundreds of thousands of identical DNA sequences that interrogate the methylation of a target location at the genome. If we zoomed in, this is a closer picture. And then when the sample DNA is added and after being amplified, it binds the complementary sequences. Upon binding, this produces a fluorescent signals through single base extension. And then these fluorescence signals are quantified to produce methylation measurements, or what we call beta values, and other measures, of course. Asking how many locations across the genome or how many CPGs are studied, this has been evolving with time, starting from the 27K array, which covers 27,000 locations, until now almost 1 million CPG by the EPIC version 2. A lot of you are familiar with the EPIC version 1, of course, which covers the 850,000 sites, and the probe overlap makes around 95%. And now let's talk about how the concept was utilized to develop the classifier, which also was touched by Jan Muller in the last session. So in Heidelberg, genome-wide methylation profiling was applied to distinguish different brain tumors based on methylation patterns of their cell of origin. So if you look at these 5 tumors from A to E, let's only focus on those 5 different CPG sites from 1 to 5, but please keep in mind that in methylation arrays, we're looking at much more. And then if you look at the methylation of them, assuming that methylated is marked with red and unmethylated is marked with blue, and please keep in mind that methylation is either yes or no, so it's either 1 or a 0. But why we have here a spectrum of color with different shades of blue and different shades of red is simply because we're talking here about bulk tumor data. So at the level of a single cell in a single gene, it's either methylated a single CPG location, it's either methylated or unmethylated. But when we study it from a tumor tissue, then we're not looking at a single cell, right? And this is why we would have a range of methylation spectrum with different shades represented here in the graph. If you look, for example, at tumor B and tumor D, you can clearly see that the patterns of methylation among different sites is closer than other tumors, for example. So this is basically because these tumors are originating from embryologically related tissues, and this is why they have closer patterns of methylation. When we plot these tumors on a 2-dimensional clustering plot, or what we call the T-SNE plot, then related tumors, B and D here, for example, they would cluster closer to each other. Beautifully, when we do DNA sequencing, then tumors with similar methylation patterns, they often also have shared mutations. This is not only limited, of course, to tumors or to CNS tumors, but you can choose different study groups and then extract DNA and then do a methylation assay. And then by quantification of methylation status at different CPG sites, you would end up identifying profiles of the different study groups. And from here, I will move to methylation epic array workflow. So as you can see, it's a simple workflow. Usually it's 3 days or 5 days according to the tissue that you're using, starting from DNA extraction in the lab, and then a bisulfite conversion step, and then the Infinium workflow, and then the scanning by the methylation scanner. If we are using FFPE material, like most of the times in majority of the sites, I'm talking here about the molecular neuropathology outreach consortium sites, which are located in low middle income setups. So mostly we're using FFPE material. So there are additional 2 steps, one of them preceding and the other following bisulfite conversion. And this makes the duration a bit longer of the workflow. And before we proceed further, I would like to stop a bit and highlight that there are 2 different platforms conducting methylation scanning at the moment from Illumina. So it's either the iScan machine, which is the standard machine that you are familiar with, but also the NextSeq 550, which is primarily a sequencer, is additionally conducting sequencing. And I'm highlighting this because in some institutions that still don't have a sequencer and are considering to procure a sequencer machine, instead of procuring a more recent sequencer, like, for example, NextSeq 1100, for example, then this older version might be a better solution because it works both as a sequencer and additionally it can conduct scanning of methylation arrays. So please keep this in your mind if you're considering to implement methylation, because this is one of the machines that has been utilized by several partners in low middle income setup. Secondly, I would focus here again on the new terminology or the IDAT file. So this is the name of the raw data file that comes from the iScan or the NextSeq. So it is the raw data file of the methylation. How to make sense of this raw data file? Of course, this can be done through downstream bioinformatics analysis, but this would need, of course, bioinformatics expertise, which is not available in all institutions, right? And it would need, of course, to have reference data and a long process. But then when we talk about CNS tumors or sarcomas, now you can utilize the classifier app, which is hosted on Heidelberg Epignostics, and which some of you are familiar with also for the last decade or so. So the beauty of the classifier is that it bypasses the needed bioinformatics analysis. And I'm mentioning this because we keep getting questions regarding we don't have a bioinformatician in our institution. This is not a problem any longer if you're using the classifier. And basically the only way to make sense of methylation data of CNS tumors or sarcomas is to compare them against the reference set. So you would need the classifier anyway. And I would just also highlight the how the interface of epignostics look like, just to show you how user-friendly the process is. So you just need to register your account. And if you are registered through an email that is acknowledged as a research partner to Epignostics, you would be allowed to upload your samples for free. And then you can just upload the two IDAT files that come from the methylation machine and just click it. And then you'll get the report within the same day. So this is really has made it so easy. And of course, you can choose the classifier version that you want.
Also, can you go back one slide? Sorry. So,
Yes, please.
no, further, one more. So this process, it seems that it takes around 3 days. So why does it take for like whenever we submit for methylation,
Yes.
it takes between 3 to 6 weeks? Is it related to the sites that we submit to and the number of samples that they have to process, or does it have to be processed in batches? Like, why does it take so long?
Yeah, this is a very good question. Yes, because part of it because of the patching, of course, because the bead chip that you see here is already designed to accommodate 8 samples at a time. In addition to that, some of the reagents that come with the kits, they are designed for 16 samples or 32 samples, and they have an expiry date of around 2 weeks. So once you open the reagent, you need to consume it within 2 weeks for 16 samples. And if you bought, for example, the larger kit, the 32 sample kit, then again, you would need to consume the reagent within 2 weeks. So you would need to run the 32 samples within 2 weeks. So it's just related to batching and which kits you are buying. And of course, when you scale up the kits, the price go lower. So the 32 sample kit is more cost effective than the 8 sample kit or the 16 sample kit. So this is the reason. But the workflow in the lab, if you're conducting methylation in a high throughput institution like Heidelberg, for example, then it would be only really the 5 day
So
or the 3 day.
Felipe and I were at a meeting in Brazil a couple of weeks ago, and there was a talk from MD Anderson about real-time methylation in the OR. And so it seems this is pretty doable because you're basically cutting the time from 3 to 5 days to within a short period of time. Because in my mind, I thought, oh, it has to take a very long time and so on, but it seems it can be done.
Yes, of course, it can be done to shorten the time, but I believe that Felipe was referring to the ONT-based algorithm. So there is the Oxford Nanopore technology algorithm, which is the newer technology than Illumina. So this whole workflow is talking about Illumina workflow, which the consortium is implementing currently at partner sites. However, we have plans to implement the Oxford Nanopore technology as well at partner sites in the future. But we need to implement one technology at a time. With Oxford Nanopore technologies, there are some advantages, of course, and disadvantages in comparison to Illumina. Illumina is the standard of care so far for methylation assays. For Oxford Nanopore technology, the biggest advantage is that you can run one sample at a time, so you don't need the batching factor. So a lot of partners who have low throughput or like small numbers of patients, then they prefer to go through the Oxford Nanopore technology. In addition to that, the protocol that has been developed in Heidelberg by Aripa Patel and other colleagues at neuropathology, it really has been well developed now to shorten the time to 30 minutes intraoperatively. So yes, thanks a lot for highlighting this development. And this really provides a lot of valuable information on time for neurosurgeons and for decisions for the patients, of course. Yes. So, yes. I think I only have a couple of two more slides that were already discussed by my colleague Jan last time. But this is just a reminder of the DNA methylation report that you are getting after you upload your IDAT files to Epignostics in order to complete the workflow. So this is a reminder of the calibrated scores as well. So less than 0.77 is generally considered no match. And there are several hierarchical outputs, the subclass, the class family, and the superfamily. And these offer more stable guidance when lower tier predictions are uncertain. Beyond the classification, Jan has also touched beautifully on TSNE and UMAPs. So for the cases which get a low score, less than 0.77, such as the cases that we basically discuss in these meetings, in addition to running the classifier, it's always beautiful to just project them against this TSNE plot. Because with the TSNE plot and the reference samples, you can infer methylation-based clusters, substructure within clusters, outlier sample groups, which might actually give a hint towards the identification of new entities. However, with TSNE plots, you cannot really infer true biological distance between clusters or lineage or evolution relationships or put a diagnostic label. This is the nice example that Jan presented last time. So if you got this sample, which has a low score for ependymoma, PF posterior fossa, and then with a low score, let's say of 0.5, for example, and then you projected it against this TSNE, and you've seen it already located near to the ependymoma PFA, then you should that gives a hint, actually, that the case might be ependymoma. So it is supportive. However, by its own, it's not diagnostic. And this is why we provide TSNE plots for those cases in the tumor boards. With that, I come to the end of this session. I thank you all for listening, and I'm open to questions, of course. But before that, I would like to thank the fantastic global partners of the Molecular Neuropathology Outreach Consortium, and of course to Mohammed, Lorena, Kim, Crystal, and all the colleagues at St. Louis. We started expanding in Europe, as you see, with the first site in Portugal. And I'm happy to have one of the new partners joining us today, Rahatul Ain. The site is not on the map. I'm sorry, Rahat, a newer site from Pakistan. And also, I would like to thank the supporters from Illumina, Epignostics, Einherz for Kinder, and Sidra Medicine. And finally, all the team in Heidelberg, especially Jan Muller, Martin Zell, Dominic Sturm, Felix Sam, David Jones, and Stefan Fester.